All right, so you sent over some pretty intense research on frizzled receptors. These proteins, they're kind of a big deal, right? They're in all these cellular signaling pathways, and they're tied to everything.
Yeah.
From like, how tissues develop and repair to even diseases like cancer.
Right.
And this paper you found, it's...
yeah, it really gets down to the nitty-gritty.
Yeah, it digs into like, how are these frizzled receptors, or we'll just call them FZDs for short, how are they activated?
That's exactly right.
Okay, so set the stage for us.
So this paper, it points out a really key difference between FZDs and this other protein called Smoothened, SMO for short.
Okay.
And both of these, they belong to this this class F group of G protein-coupled receptors.
Right. Those are like the big communicators inside cells.
Yeah, exactly. Those crucial signal transmitters.
Mm-hmm.
But the way they get activated, that's where things get interesting. There are some pretty intriguing differences.
Okay, so the paper really focuses on this thing called P6.43. What is that all about? What's so special about P6.43?
Well, it all boils down to just one single amino acid.
Okay.
At this specific spot, position 6.43 in their structure, FZDs have this proline amino acid, while SMO has a phenylalanine.
Okay.
Now, this might seem like a tiny little difference.
Right, like who cares? Swap them out. What's the big deal?
Right. But you'd be surprised, this tiny difference actually has huge implications.
Interesting.
Yeah, for both their shape and how they function.
Mm-hmm.
So proline, with its unique structure, it actually creates this kink in the FZD protein.
A kink, like a bend in the road.
Exactly, like a bend in the road.
Okay.
SMO, on the other hand, with its phenylalanine at that spot, it stays pretty straight.
Okay, so one's got a curve, one's a straight line. Got it.
Exactly. And this shape difference, this is what affects how they interact with other molecules.
Got it. Okay.
Yeah.
So then the paper brings up cholesterol.
Mm-hmm.
And it's saying that SMO needs cholesterol to function, but STDs don't. I'm guessing this ties back to that whole shape difference we were just talking about.
You got it. Because SMO has that straight structure, it can actually form this little pocket, a perfect fit for cholesterol.
Like a key in a lock.
Yeah, exactly like a key in a lock.
Okay.
And this binding, the cholesterol fitting in there, it's essential for SMO to become active.
I've got it.
But FZDs, with their kink, they don't have that pocket.
Right.
So they've evolved this different mechanism, a way to activate that doesn't need cholesterol binding.
Fascinating.
Yeah.
Okay, so now this is where things start to get really complex.
Mm-hmm.
The paper dives into this thing called a molecular switch.
Right.
And these networks of aromatic amino acids.
Mm-hmm.
So first of all, what are these aromatic networks?
Okay, so imagine this molecular switch like a tiny little lever.
Okay.
It controls whether the FZD is on or off, right?
Got it.
And this switch, it's held in place, stabilized by this network of aromatic amino acids. These amino acids, they have these unique ring-like structures, and they can interact with each other in a very specific way, forming this intricate scaffold.
Like a support system for that little lever.
Precisely. Yeah, it keeps the switch in place.
Got it.
Yeah.
And so this aromatic network, is that different in FZDs compared to SMO?
Yes, that's exactly it.
Okay.
FZDs have a more extensive aromatic network, probably because of that kink in their structure.
Okay, so the kink kind of gives it more surface area.
They got it.
For those interactions to happen.
Yeah, for those aromatic interactions to occur, creating this more elaborate and robust switch mechanism.
Interesting.
Mm-hmm.
Okay, so we've got this shape difference.
Mm-hmm.
We've got the whole cholesterol binding thing and this molecular switch going on.
Yeah.
But how does all this actually translate into, like, real-world function?
Right.
What does this mean for how these proteins actually do their jobs within a cell?
That's a great question. And that's where the researchers, they took it a step further. They actually did these experiments where they mutated that key proline in FZD6, changed it to a phenylalanine.
So they were basically trying to straighten out that kink.
Yeah, essentially trying to make it more like SMO, see what happens.
Okay, so they're like, let's swap their outfits and see if they can still dance.
Exactly. Exactly. And what they found was pretty amazing. This mutation significantly reduced FZD6's ability to bind to this other protein called DVL2.
DVL2, okay. And why is that important?
DVL2 is a central player in the want signaling pathway.
Okay, remind me again, what's the want signaling pathway?
Think of it as a crucial communication system within cells.
Okay.
It's involved in all sorts of processes, from embryonic development to tissue regeneration. It's a busy pathway.
Right, so it's kind of a big deal.
Yeah, it's a big deal, and DVL2 is a key part of that whole communication network.
Okay, so by changing that one amino acid, the researchers basically disrupted the communication lines between FZD6 and DVL2.
That's exactly it. They messed up the communication. And that disruption had these significant downstream effects, changing the way FZD6 could activate the whole white signaling pathway.
Wow.
Yeah.
But wasn't there something else? Didn't the mutation also affect how FZD6 interacts with another pathway?
Yeah, that's where it gets even more interesting. The mutation didn't affect FZD6's ability to activate this other pathway, one that works through a protein called Gs.
Okay.
So it's not like a simple on-off switch.
Right. It's more nuanced.
Yeah, much more nuanced.
Oh.
Mutating that one amino acid had these selective effects, impairing one pathway while leaving the other one completely intact.
Wow. So it's not just about whether FZD6 is on or off.
Right.
It's about which pathways it can activate and how those pathways are being affected by these, like, subtle structural changes.
Exactly.
This is pretty mind-blowing stuff.
It is, but that's what makes this research so fascinating.
Yeah.
It shows just how intricate and fine-tuned these cellular processes really are, and how even the tiniest change, like swapping one amino acid, can have this ripple effect throughout the entire system.
Okay, so to really drive that point home, the researchers also did the reverse experiment, right?
Yes, they did.
They took SMO, and they mutated its phenylalanine to a proline.
Yep.
So basically, they tried to force a kink where there wasn't one.
Exactly. See if they could make SMO more like FZD.
Right. Okay, and what happened?
And guess what? It basically shut down SMO's activity almost completely.
Wow, so both proteins are super sensitive to changes at that one specific spot.
They are. It's incredible.
Okay, so we've got these two proteins, FZDs and SMO, with their unique structures, their own ways of being activated.
Yeah.
And we're starting to see how those tiny little details at the molecular level can have these huge ripple effects on much larger cellular processes. But remember, we've been focusing on FZD6 mainly here. And the FZD family, it's actually pretty diverse.
Oh, it is. There's a whole bunch of FZDs, each with its own little quirks, variations on the theme.
Didn't the paper point out that the effects of that proline mutation, they weren't the same across all the FZDs?
Exactly. They found that mutating the proline in some FZDs had a much bigger impact than in others.
Interesting.
Yeah, really highlighting the diversity within this family of proteins.
Okay, so we've got this intricate world of FZD receptors, each with its own unique structure, its own way of being activated, and they're all playing these crucial roles in these cellular signaling pathways. Now, before we get too lost in all the molecular details, let's take a step back. Why does this even matter? Why should our listener care about all this molecular mumbo-jumbo?
Well, think about it. We're talking about processes that are fundamental to how our bodies develop, how we stay healthy, and even how diseases like cancer progress.
Right.
Understanding these intricacies of proteins like FZDs and SMO is crucial for developing therapies that actually work.
So we're talking targeted therapies, right?
Exactly. Therapies that can actually hit the faulty processes without causing a ton of side effects.
So it's like having a scalpel instead of a sledgehammer.
That's a perfect analogy, a scalpel instead of a sledgehammer.
Okay.
And that's what makes this kind of research so exciting.
So by understanding these subtle differences in how FZDs and SMO are activated, we can start to develop drugs that can target one or the other.
Yes.
Potentially leading to more effective treatments.
Right.
With fewer side effects.
That's the ultimate goal.
Wow, that is exciting.
It is.
Okay, so this is already blowing my mind, and we've learned about these differences between FZDs and SMO, how they're activated, why understanding these molecular details is so important for developing new treatments. But this is just the tip of the iceberg, right?
Oh, absolutely. This paper, it raises just as many questions as it answers.
Yeah.
And that's what makes this field so fascinating.
Well, then let's dive even deeper.
Let's do it.
Are you ready to explore some of those unanswered questions and see where this research might lead us?
Always. The complexities of cell signaling are endlessly fascinating.
That's the spirit. Let's unravel some more of these molecular mysteries.
I'm ready.
Okay, so we've established that FZDs and SMO, they've got these distinct activation mechanisms.
Right.
But the paper, it also hints at something even more intriguing, different, like activation pathways.
Yes.
What does that even mean? And how does it relate to all this talk about potential therapies?
That's a really crucial point.
Okay.
It's not just about whether an FZD is on or off, right?
Right.
It's about how it's activated and what affects that activation triggers.
Okay.
You see, SMO, it's relatively straightforward.
Okay.
It mainly activates this thing called the hedgehog pathway, which is crucial for development.
Right.
but it's also implicated in some cancers.
Okay, so SMO is like a specialist focused on one particular job.
Yes, exactly.
But FZDs, they're more like multi-talented performers.
Precisely.
Okay.
FZDs can activate a variety of pathways, including the Wnt-catenin pathway that's essential for embryonic development and cell growth.
Okay.
And then there's the Wnt-planar cell polarity pathway, or PCP pathway for short.
Okay.
That one helps cells orient themselves within tissues, making sure everything's lined up correctly.
So, like, it makes sure everything is in the right place.
Exactly. Keeps everything organized.
Okay, so we've got these two major pathways, Wnt-catenin and bundle TPCP, both influenced by FZDs.
Mm-hmm.
But how do FZDs decide which pathway to activate? It's not like they just flip a coin, right?
That's where it gets really fascinating.
Okay, tell me more.
The paper suggests that FZDs can actually change their shape.
Really?
Yeah. Adopt different shapes or conformations to interact with specific partners and activate specific pathways.
Right.
It's kind of like a key fitting into a lock,
Right.
Only certain keys can unlock certain doors.
Okay, I get it.
Similarly, different FZD shapes or conformations can trigger different downstream signaling events.
So it's not just about whether the FZD is on, it's about its specific shape.
Yes.
Which then determines which pathway it activates.
Yeah.
Okay, so that molecular switch we talked about earlier, that probably plays a role here, right?
Absolutely. The way that molecular switch is flipped, influenced by those aromatic networks we discussed,
Right.
It seems to dictate which partners the FZD can interact with and which pathways it can trigger.
This is like a complex dance with FZDs changing partners and steps to create different dances, each with its own unique outcome.
That's a great analogy, a beautiful dance. And remember, all of this is influenced by those subtle structural differences.
Right, the proline kink versus the straight phenylene.
Exactly, tiny changes with huge consequences.
Okay. Before my head explodes, let's bring this back to real-world applications.
Right.
How does understanding these different activation pathways help us develop better drugs?
Imagine being able to design drugs that not only target specific FZDs, but also fine-tune their activation, nudge them towards one pathway or another. That's the power of understanding these nuances.
So instead of just blocking FZD activity altogether,
right,
which could have a lot of unwanted side effects.
Exactly.
We could be more precise.
Yes.
It's like composing a specific melody instead of just hitting a random note on a piano.
That's a great way to put it.
Okay.
And that's why this research is so important. It's paving the way for these more effective therapies with fewer side effects. It could revolutionize how we treat diseases.
That's incredible.
It is.
Okay, this deep dive is already blowing my mind.
Good, good.
We've learned about these crucial differences between FZDs and SMO, how they get activated,
right,
and why understanding these molecular nuances is so important for developing new treatments.
Yeah.
But what else did the researchers uncover about the functional consequences of that key amino acid difference between FZDs and SMO?
They dug deeper into how those mutations, swapping that proline for a phenylalanine, affected the protein's ability to bind to other key players in those signaling pathways. And the results were, they were pretty revealing.
Okay, give me the highlights. What did they find?
Remember DVL2, that crucial component of the Wnt signaling pathway?
Yeah.
Well, when they mutated FZD6 to make it more like SMO, its ability to bind to DVL2, it was significantly reduced.
So that tiny change, it really messed things up.
It did. It's like cutting a vital wire in the cellular communication network.
Wow.
But here's where it gets even more interesting. The mutation didn't completely shut down FZD6.
Okay.
It could still activate another pathway, the one involving the G's protein.
So it's not a simple on-off switch.
No, it's not.
Mutating that one amino acid affects different pathways in different ways.
Precisely.
Okay.
And that's a key takeaway here. These proteins, they're incredibly complex.
Yeah.
They have multiple facets to their function. It's not just about whether they're active or inactive.
Right.
It's also about which partners they can interact with and which pathways they can trigger.
Okay, so we're dealing with a whole network of interactions here.
We are.
Each one finely tuned by these subtle structural differences.
That's right.
Okay.
And that's what makes understanding these molecular details so important for developing those targeted therapies, right? We need to know not only which proteins to target,
right,
but how to actually manipulate their interactions in a precise and controlled way.
This is like playing 3D chess with molecules.
It really is. It's a challenging but incredibly rewarding field.
Yeah.
And with each new discovery, we're getting closer to unlocking the secrets of how our cells communicate.
I'm ready for another layer of complexity.
Bring it on.
What else did the researchers uncover about how these mutations affect FZD function?
They took a closer look at this specific part of FZD6 called the DEP domain.
Okay.
This domain, it's known to interact with other proteins, particularly one called DVL, which is like a central hub in those want signaling pathways we were talking about.
Okay, so by studying this DEP domain, they could get even more insight into how those mutations affect CD6's ability to engage with its partners.
Okay.
And what they found was fascinating.
What they find?
The mutation that made FZD6 more like SMO, that proline to phenylalanine swap, it actually altered its ability to recruit the DEP domain.
So even though FZD6 wasn't completely shut down, this mutation was still messing with its ability to interact with those key partners in the want signaling pathway.
Exactly.
Okay.
And this finding, it really reinforces the idea that these subtle structural differences, they have huge consequences for how these proteins function. It's not just about turning them on or off.
Right.
It's about fine-tuning their interactions to make sure the right signals get sent at the right time.
This is like a delicate dance.
Yes.
With those tiny amino acid changes acting as choreographers.
I love that analogy.
Dictating which partners the proteins can dance with.
That's beautiful. It highlights the incredible complexity and elegance of how our cells communicate. It's a symphony of molecular interactions, each playing a crucial role in orchestrating those processes that keep us alive and functioning. Okay, I'm starting to feel like I'm conducting this molecular orchestra.
I like it.
But let's zoom out for a second.
Okay.
Why does all this matter? How does understanding these intricate details help us develop better treatments for diseases?
That's the ultimate goal, isn't it? And the implications, they're huge.
Yeah.
By understanding how these proteins interact and how those interactions are affected by even the tiniest changes.
Right.
We can start to design drugs that target specific pathways with incredible precision.
So we're talking about drugs that can not only hit the right target,
yes,
but also fine-tune the cellular response.
Exactly. Instead of just blocking a protein altogether, which could have all sorts of unintended consequences,
right,
we could develop drugs that act like molecular choreographers.
I like that.
Guiding those interactions to achieve the desired therapeutic effect.
That's pretty mind-blowing.
It is.
So this paper, with its focus on a single amino acid difference, it's actually a big step towards developing this whole new generation of targeted therapies.
Absolutely. It's a testament to the power of basic research, of delving into those fundamental molecular mechanisms to uncover new avenues for treating diseases.
And that's what makes this field so exciting.
It is. Every discovery has the potential to transform lives.
Okay, I'm officially hooked on this molecular detective story.
Good.
What else did the researchers uncover about the functional consequences of that key amino acid difference?
Well, remember how we discussed that FZD6, unlike SMO, it can activate different signaling pathways. They wanted to explore whether that key amino acid difference played a role in this pathway selectivity.
So they were trying to figure out if that proline kink in FZD6 was somehow influencing which pathway it decided to activate?
Exactly.
Okay.
And to do this, they used this clever assay called the TOPflash assay.
Okay.
It's basically a way to measure the activity of a specific pathway, the one-catenin pathway, which is involved in all sorts of cellular processes, from development to cancer.
So they could use this assay to see how well different FZDs, with and without that proline mutation, could activate this particular pathway.
Precisely. Yeah. And the results were pretty striking. They found that the mutation that made FZD6 more like SMO, that proline to phenylalanine swap, it actually reduced its ability to activate the one-catenin pathway.
So that tiny change was enough to shift FZD6's allegiance away from one of its usual pathways.
That's what the data suggests.
Wow.
Yeah, it highlights the importance of that proline kink in maintaining FZD6's ability to engage with specific partners and activate specific pathways.
This is getting really complex.
Yes.
It's like that one amino acid is a master puppeteer pulling the strings to control which pathways get activated.
And that's the beauty of it, isn't it?
Yeah.
These seemingly small changes at the molecular level can have these cascading effects on much larger cellular processes.
Right.
It's a reminder that even the tiniest details matter.
Okay, before we spiral too deep into the molecular abyss,
right,
let's bring this back to our listener. Why should they care about all this? What's the big takeaway?
Well, this research, it really underscores the incredible complexity and elegance of how our cells communicate. It highlights how proteins like FZDs and SMO, these tiny molecular machines
Yeah.
are constantly interacting and changing shape to orchestrate the processes that keep us alive and functioning.
So we're talking about this whole universe of molecular activity happening inside us,
We are.
all guided by these subtle structural differences and intricate interactions.
That's right.
Wow.
And the more we understand about this molecular universe, the better equipped we'll be to develop therapies that can actually target specific pathways and treat diseases with greater precision and fewer side effects.
Okay, I'm officially awestruck by the intricate workings of our cells.
Good, good.
But I feel like we've only just begun to explore the depths of this research.
Oh, we have, absolutely.
There's still so much we don't know, right?
Absolutely. This caper is just one piece of the puzzle, one glimpse into the vast and complex world of FZD receptor activation.
Oh.
There's still so much we don't know. But each discovery brings us closer to unlocking those secrets of cellular communication
Right.
and harnessing that knowledge to improve human health.
Well, then let's keep diving deeper.
Let's do it.
What other fascinating insights did the researchers uncover about those different activation pathways and how they're regulated?
They actually discovered some really intriguing differences in how those pathways are activated, depending on the specific FZD involved.
Okay.
And it all ties back to those subtle structural variations we discussed earlier.
Okay, spill the details. I'm eager to learn more about how those structural differences play out in the real world of cellular signaling.
One of the key findings was that not all FZDs rely on that proline kink to the same extent.
Oh.
Remember how we talked about FZD7?
Yeah, the one that can activate that G's protein pathway.
Exactly. Well, it turns out that FZD7's activity wasn't really affected by mutating that key proline to a phenylaline.
So unlike FZD6, which seemed pretty sensitive to that change,
Right.
FZD7 just kind of shrugged it off.
That's what the data suggests.
Interesting.
Yeah, and this finding highlights the diversity within the FZD family.
Okay.
Even though they share some structural similarities, they're not all identical twins.
Right.
More like cousins with distinct personalities.
I like that.
Yeah.
So even though that proline kink seems to be important for some FZDs, it's not a one-size-fits-all rule.
Exactly.
Okay.
And that's an important nuance to keep in mind.
Yeah.
It means that we can't just assume that all FZDs will behave the same way.
Right.
We need to study each one individually to understand its unique properties and how it contributes to those complex signaling pathways.
This is making my brain do some serious gymnastics.
I know, it's a lot to take in.
But I'm loving the challenge.
Good, good.
So I've got this diverse family of FZDs,
Yes.
each with its own quirks and sensitivity.
That's right.
And remember, we're only talking about a handful of FZDs here.
Right.
There are 10 in total.
That's right.
Each potentially involved in multiple pathways and interacting with a whole cast of other proteins.
It's a lot of characters to keep track of.
It is. It's a complex and dynamic network.
It is.
Endless possibilities for exploration.
Absolutely.
Okay, let's zoom in on one specific pathway for a moment.
Okay.
The paper mentioned something about the one-planar cell polarity pathway, or PCP, being particularly sensitive to that proline mutation.
Yes, the PCP pathway, it's all about how cells orient themselves within a tissue, like little compasses, making sure everything's lined up correctly.
Yeah.
And it seems that that proline kink in FZD6, it's crucial for its ability to properly activate this pathway.
So when they mutated that proline to a phenylaline, it messed with FZD6's ability to direct those cellular compasses.
That's what the data suggests.
Wow.
Yeah, and this finding has some really interesting implications for understanding how tissues develop and how disruptions in those developmental processes can lead to diseases.
Okay, I'm intrigued. How does this relate to tissue development and disease?
Imagine if those cellular compasses get thrown off kilter.
Okay.
Instead of lining up neatly, the cells start to point in different directions.
Oh.
Yeah, creating chaos and disorganization within the tissue.
So it's like a construction project gone wrong, with the bricks being laid haphazardly instead of following a blueprint.
That's a great analogy.
Okay.
And this kind of disorganization, it can have serious consequences, potentially leading to developmental defects or even contributing to the progression of diseases like cancer.
Wow, so that tiny proline kink is playing a big role in ensuring that our tissues develop correctly and stay organized.
It is. It highlights the importance of understanding these subtle structural differences at the molecular level. They can have these ripple effects on much larger processes, ultimately influencing our health and well-being.
Okay, I'm officially blown away by the interconnectedness of it all.
Good, good.
But before we get too lost in the cellular weeds, let's bring this back to our listener.
Okay.
Why should they care about the PCP pathway and how it's regulated?
Think about it. The PCP pathway is crucial for all sorts of developmental processes, from the formation of our organs to the wiring of our nervous system. And when this pathway goes awry, it can contribute to a wide range of disorders.
So we're talking about birth defects, neurological problems, maybe even cancer.
Exactly.
Wow.
And by understanding how proteins like FZD6 regulate this pathway and how those regulatory mechanisms can be disrupted, we can start to develop new strategies for preventing and treating these conditions.
Okay, I'm seeing the big picture now.
Good.
This research isn't just about understanding the intricacies of cellular communication. It's about translating that knowledge into real-world solutions for improving human health.
That's the ultimate goal.
That's incredible.
It is. It's a testament to the power of basic research.
Okay.
By delving into those fundamental molecular mechanisms, we're uncovering new avenues for understanding and treating diseases.
All right. I'm ready to keep exploring those avenues.
Let's do it.
What else did this paper reveal about the PCP pathway and how it's affected by that key amino acid difference?
One of the key findings was that not all FZDs rely on that proline kink to the same extent when it comes to activating the PCP pathway.
Okay.
Remember how we talked about FZD5?
Yeah, wasn't that one of the FZDs that was less affected by that proline to phenylalanine mutation when it came to activating the 1-to-catenin pathway?
Exactly. And it turns out that FZD5 is also less reliant on that proline kink for activating the PCP pathway.
So it's like FZD5 has its own unique way of doing things.
Yes.
Regardless of whether it has a proline or a phenylalanine at that key position.
That's what the data suggests.
Fascinating.
Yeah, and this finding further reinforces the idea that we can't just assume that all FZDs will behave the same way.
Right.
We need to study each one individually to understand its unique properties and how it contributes to those complex signaling pathways.
This is making me appreciate just how diverse and adaptable the FZD family is.
It is.
Each member seems to have its own special talents and sensitivities.
That's a great way to put it, and that diversity is likely what allows FZDs to play such a crucial role in so many different cellular processes.
Right.
They're like a team of specialists, each equipped to handle a specific task.
Okay, I'm loving this analogy of the FZDs as a team of cellular specialists.
Sure.
But let's bring it back to our listener for a moment.
Okay.
Why should they care about the fact that some FZDs are more reliant on that proline kink than others? What's the big takeaway here?
Well, this finding has some important implications for drug development.
Okay, how so?
It means that we can't just develop drugs that target all FCEs equally.
Right.
We need to be more precise, taking into account the unique properties of each FZD and how it interacts with specific pathways.
So it's like developing a personalized medicine approach.
Yes.
Tailoring the treatment to the specific FZD involved in the disease.
Exactly. And that's the beauty of this kind of research. It's laying the groundwork for a whole new generation of targeted therapies, therapies that are more effective and have fewer side effects.
Okay, I'm officially on board with this precision medicine revolution.
Good. Good.
But before we get too carried away with future possibilities, let's circle back to the research itself.
Okay.
What were some of the limitations or challenges the researchers faced in studying these complex FZD interactions?
Well, one of the biggest challenges is that we're still missing a complete picture of how FZDs work. We have these snapshots, these glimpses into their interactions.
Right.
But there's still so much we don't know.
So it's like trying to solve a puzzle with only a few pieces.
That's a great analogy.
Okay.
And it means we have to be cautious about drawing definitive conclusions from any single study.
Right.
We need to keep gathering data, keep piecing together the puzzle to gain a more comprehensive understanding of these complex signaling pathways.
Okay, so it's a work in progress, but an incredibly exciting one.
It is. It is.
What were some of the specific challenges the researchers faced in this particular study?
Well, one challenge was simply the sheer complexity of the system they were studying. Okay. We're talking about multiple proteins interacting in dynamic ways. And it's difficult to capture all those interactions in a single experiment.
So it's like trying to film a dance with 1,000 dancers.
Yeah.
Each moving independently, but also influencing the movements of others.
Exactly.
Wow.
And it requires sophisticated techniques and careful experimental design to tease apart those individual movements and understand how they contribute to the overall choreography.
Okay, I'm starting to feel like a molecular choreographer myself.
Ah, I like it.
But I'm also getting a sense of the challenges involved in studying these complex cellular processes.
And those challenges, that's what makes this research so rewarding.
Yeah.
Every new insight, every piece of the puzzle we uncover, it brings us closer to understanding the language of life itself.
That's a beautiful way to put it.
Thank you.
Okay, I'm ready to delve even deeper into this molecular dance.
Let's do it.
What other challenges did the researchers encounter in studying those FZD interactions?
Well, one challenge they highlighted was the difficulty of studying these proteins in their natural environment within living cells.
So it's one thing to study these proteins in a test tube.
Right.
But it's a whole other challenge to see how they actually behave within the complex and dynamic environment of a cell.
Exactly. That's why the researchers used a variety of techniques, including some really clever imaging methods, to try to capture those real-time interactions within living cells.
So they were basically trying to spy on those molecular dancers in their natural habitat.
That's a great way to put it. And by doing so, they were able to gain some really valuable insights into how those proteins behave in a more realistic setting.
Okay, I'm starting to feel like a molecular detective, piecing together clues from these different experiments to build a case for how these proteins work.
I like it.
But I'm also realizing that we're still missing some key evidence, right?
Oh, absolutely. This research, it's just the beginning. We still need more studies, more data to fill in those gaps and gain a truly comprehensive understanding of FZD receptor activation.
Well, then let's keep investigating.
Let's do it.
What were some of the specific questions that the researchers left unanswered in this paper? What are the mysteries that still need to be solved?
One of the big mysteries is how those different FZD conformations, those different outfits we talked about, actually translate into different signaling outcomes.
Okay.
We know that they can adopt different shapes.
Right.
But we don't fully understand how those shapes dictate which pathways get activated.
So it's like we know the dancers have different costumes, but we don't know the choreography.
That's a great analogy, and it's one of the key questions that future research needs to address.
Okay.
We need to crack the code.
Right.
Understand how those structural changes translate into specific signaling instructions.
Okay, I'm adding that to our molecular mystery list.
Good.
What other unanswered questions did the researchers highlight?
Another big question is how those FZD interactions are regulated in different cell types and tissues. Okay. We know that they play a role in a wide range of processes, from development to cancer, but we don't fully understand how those roles vary depending on the cellular context.
So it's like the same dance can be performed differently in different venues.
Exactly, and it's important to understand those variations because they could have implications for developing targeted therapies.
Right, because what works in one cell type might not work in another.
Precisely.
Okay, I'm starting to see just how much more there is to discover about these fascinating proteins.
Oh, there's so much more.
But I also feel like we've made some significant progress in our deep dive today.
We have.
We've learned about the crucial differences between FZDs and SMO, how those differences translate into different signaling outcomes,
right,
and why understanding these molecular nuances is so important for developing new treatments.
Yeah, absolutely. And remember, we're only scratching the surface here. There's a whole universe of molecular interactions happening within our bodies, and each discovery brings us closer to understanding the language of life itself.
Okay, before we get too lost in the cellular cosmos, let's bring this back down to earth for our listener.
Okay.
What's the key takeaway message from all of this? Why should they care about the complexities of FZD receptor activation?
Well, this research, it highlights the incredible power of basic science, of delving into those fundamental molecular mechanisms to uncover new avenues for treating diseases.
Right.
It's a reminder that even the tiniest details matter.
Yeah.
And that every discovery has the potential to transform lives.
That's a powerful message, and it's testament to the dedication and ingenuity of the researchers who are tirelessly working to unlock the secrets of our cells.
Absolutely, and we're just at the beginning of this journey. There's still so much to explore, so much to discover, and the possibilities for improving human health are truly limitless.
Well, then let's keep exploring those possibilities.
Let's do it.
What other fascinating insights did the researchers uncover about those different FZD activation pathways and how they're regulated?
They actually made a really intriguing observation about how cholesterol, that fatty molecule we often hear about in relation to heart health, might be playing a role in FZD activation.
Wait, cholesterol? I thought we established that FZDs don't need cholesterol to function, unlike SMO.
That's right. They don't need it in the same way that SMO does.
Right.
Remember how SMO has that special pocket where cholesterol binds and activates the protein? FZDs don't have that pocket.
Okay, so if FZDs don't have that binding site, How could cholesterol be involved?
Well, the researchers, they noticed that even though FZDs don't directly bind cholesterol within their core structure, cholesterol seems to be influencing their activity in a more indirect way.
Okay, I'm intrigued. How does that work?
Think about it this way. Cholesterol is a key component of cell membranes.
Those outer layers that surround our cells.
Exactly. And since FZDs are embedded within those membranes, their activity can be influenced by the properties of the membrane itself.
So it's like the stage can affect the performance of the dancers, even if the dancers don't directly interact with the stage.
That's a great analogy, and cholesterol is one of the key players in determining the fluidity and organization of those cellular stages.
Okay, so this is where the research, it expands beyond just the cell membrane itself.
Okay.
It starts to look at how the environment around FZDs might be influencing their activity.
We're looking beyond the individual dancers and the stage to consider the whole theater,
Exactly.
the lighting, the set design, maybe even the audience.
Precisely. And one crucial environmental factor is this thing called the extracellular matrix, or ECM for short.
ECM, got it.
This is it's a complex network of proteins and other molecules that surround our cells.
Okay.
It provides structural support, but it also regulates a whole bunch of cellular processes.
So the ECM is like the architecture of the theater.
Yes, exactly. Influencing how the FZD dancers can move and interact.
Precisely. The ECM, it can interact with FZDs directly or indirectly.
Okay.
It can even bind to them, actually, and modulate their activity.
So it's as though the scaffolding of the theater has like special handles or pathways that the dancers can use, influencing their performance.
That's a great way to visualize it.
Okay.
And these interactions between the ECM and FZDs, they can have a big impact on which signaling pathways are activated and how strong those signals are.
This adds a whole other level of complexity to the FZD story.
It does.
But why should our listener care about the ECM and its impact on FZDs?
Because this opens up the possibility of actually targeting the ECM as a way to treat diseases that involve FZD dysfunction.
Okay.
Imagine being able to remodel the theater, so to speak, to create a more favorable environment for the FZD dancers, optimizing their performance.
So we might be able to develop therapies that don't directly target FZDs themselves.
Right.
But instead modify the ECM around them.
Exactly.
That's a whole new approach.
Yeah, it is. And it highlights the importance of understanding the cellular context, not just the individual molecules, when we're developing new treatments.
Of course, studying something as intricate as the ECM, that comes with its own set of challenges, right?
Oh, absolutely. The ECM is incredibly complex, and it's constantly changing. It's made up of hundreds of different molecules all interacting and influencing each other. So isolating specific effects, it's a major hurdle.
It's like trying to study the architecture of a building during an earthquake.
Ooh, I like that.
With everything shifting and rearranging.
That's a very vivid analogy. It takes incredible precision and ingenuity to tease apart those individual elements of the ECM and understand their roles in FZD regulation.
This has been an incredible deep dive into the world of FZDs.
It has.
We've gone from the structure of a single amino acid to the complexities of the cellular environment.
Yeah.
All to understand how these tiny molecular machines can have such a huge impact on our health.
And this is just the beginning. There are still so many questions to answer, so many mysteries to unravel.
Before we let our listener go, what's the one big thought you want to leave them pondering? What's the most mind-blowing takeaway from all of this?
Think about this. If we can truly understand the steps of this intricate molecular dance, the possibilities for manipulating these pathways are limitless. We could treat diseases in entirely new ways, potentially even enhance our own development.
Wow.
All by fine-tuning these cellular signals.
That's a powerful thought to end on, is like we've been given a glimpse behind the curtain of life itself, and now we're starting to understand the choreography.
I love that.
Thanks for guiding us through this incredible journey.
It was my pleasure. Always happy to dive deep into the molecular world.
And to our listener, keep those brains buzzing, keep asking those questions, and keep exploring the fascinating world of science. Until next time, stay curious.